don't you all fade away. Don't try to dig what we all say. About my generation. I'm trying to cause a big sensation. About my generation. Talking about my generation. Talking about my generation. My generation. My generation, baby. Don't you all fade away. Don't try to dig what we all say. About my generation. I'm not trying to cause a big sensation. Just talking about my generation. My generation, baby. My generation. People try to put us to down just because we get around. Things ain't too awful. Oh, I hope I die before I get old. My generation, baby. My generation.
Since I got you on my mind, and now you are here, I said it's okay to see what we can do, baby. Just me and you, come on and stir it up. Little darling, stir it up. Come on and stir it up. Little dog. shot the sheriff, but I didn't shoot no deputy. Oh no, oh, I shot the sheriff, but I didn't shoot no deputy. Ooh, ooh, ooh. Yeah, all around in my hometown, they trying to track me down. Yeah, they say they want to bring me in guilty for the killing of a deputy, for the life of a deputy. But I say, oh now, oh, I shot the sheriff, but I swear it wasn't self-defense. Ooh, ooh, ooh. Yeah, I said I shot the sheriff, oh Lord, and they say there's a capital offense. Ooh, ooh, ooh. Yeah, hear the Sheriff John Brown always hated me for what? I don't know. Every time I plant a seed, he said kill it before it grows. He said kill them before they grow. And so, read it in the news. I shot the sheriff, but I swear it wasn't self-defense. Where was the deputy? I say I shot the sheriff, but I swear it was in self-defense. Yeah. Freedom came my way one day, and I started out of town, yeah. All of a sudden, I saw Sheriff John Brown aiming to shoot me down. So I shot him down. And I said, if I am guilty, I will pay. I shot the sheriff, but I say, but I didn't shoot no deputy. I didn't shoot no deputy. No. I shot the sheriff, but I didn't shoot no deputy. Oh, ooh, ooh, ooh. Yeah. Reflexes are the better of me, and what is to be must be. Every day the bucket I go a well. One day the bottom I go drop out. One day the bottom I go drop out. I say, I shot the sheriff. Lord, I didn't shot the deputy. No. Yeah, I shot the sheriff, but I didn't shoot no deputy. Yeah,
Microphone check, microphone check. Check, mic. Mic number 2, check, 1, 2.
So slashed and torn.
sail away, find serenity. Oh, the canvas can do miracles. Just you wait and see, then you believe me. Sailing takes me away to where I've always needed to be. Just a dream in the wind to carry me. Soon I will be
shot the sheriff, but I didn't shoot no deputy. Oh no, oh, I shot the sheriff, but I didn't shoot no deputy. Ooh, ooh, ooh. Yeah, all around in my hometown, they're trying to track me down. Yeah, they say they want to bring me in guilty for the killing of a deputy, for the life of a deputy. But I say, oh now, oh, I shot the sheriff, but I swear it wasn't self-defense. Ooh, ooh, ooh. Yeah, I said I shot the sheriff, oh Lord, and they say there's a capital offense. Ooh, ooh, ooh. Yeah, hear this? Sheriff John Brown always hated me. For what? I don't know. Every time I plant a seed, he said kill it before it grows. He said kill them before they grow. And so, read it in the news. I shot the sheriff, but I swear it wasn't self-defense. Where was the deputy? I say I shot the sheriff, but I swear it was in self-defense. Yeah. Freedom came my way one day, and I started out of town, yeah. All of a sudden, I saw Sheriff John Brown aiming to shoot me down. So I shot him down. And I said, if I am guilty, I will pay. I shot the sheriff, but I say, I didn't shoot no deputy. I didn't shoot no deputy. No. I shot the sheriff, but I didn't shoot no deputy. Oh, ooh, ooh, ooh. Yeah. Reflexes are the better of me, and what is to be must be. Every day the bucket I go a well. One day the bottom I go drop out. One day the bottom I go drop out. I say, I shot the sheriff. Lord, I didn't shot a deputy. No. Yeah, I shot the sheriff, but I didn't shoot no deputy. Yeah, so yeah.
Good afternoon. Those are really bright lights. This is fun. Welcome back. People are slowly trickling in. For you folks online, I'm sure you were very prompt. We apologize for the delay in starting, but I think we're going to stay on schedule. I think we're going to be fine. So I wanted to welcome you back to the second session of the Spring Clinical Trials Training course. In this practicum session, we're going to cover the exercises in the CTTC manual you received this morning in pages 33 through 68. For our virtual participants, these were posted to the SWOG website for you to download or print. The purpose of this practicum is to provide you with a good foundation and understanding of the protocol policies and procedures you will encounter as a SWOG Clinical Research Associate, which we call CRA. Please note that we use the term CRA very broadly. In this practicum, CRA means anyone responsible for submitting data for SWOG protocols. For this practicum session, we have presenters on site and virtually from the Data Operations Center in Seattle, so yay us for making the commute. We're going to go through the exercises with you and answer any questions you may have. For those of you not local, I want you to know this weather is a fluke. It rains every other day. You do not want to move here. Please ask questions at the microphones here if you're in person in the center aisle as time allows. Those participating virtually, we are monitoring the chat session and it's been very quiet all day. So either we're extremely thorough and have answered any curiosities you have, or maybe you should pay more attention. I'm not here to judge, I'm just saying we'd love to hear any questions you have because you do have a panel of quasi-experts here and why not take advantage of it? I doubt it, but if we end up with too many questions, we'll take note of these and send out answers by email to all attendees once we return to the office. It's important to note that due to circumstances around the pandemic and holding a hybrid meeting with remote and in-person attendees, the training you received today is different than the practicum available on the SWOG website at www.swog.org. Our aim is to give you a practical understanding of the importance of correct and complete documentation of each patient's experience on a SWOG clinical trial. Accurate and timely data submission is essential in fulfilling the objectives of any study, as we have mentioned a few times this morning. We will work through a number of exercises that involve verifying eligibility, filling out forms, conducting disease assessment, identifying adverse events, and documenting the patient's experience on SWOG forms. We have 4 presenters from the Data Operations Center who will facilitate at the podium. Myself, Christine Magner, but we'll get to know each other later. Leah Everhart, Tracy Marr, and Larry Kay. The correct answers for each exercises are pre-filled in your manual and located at the bottom of the page in very small type. Additional information about each topic can be found in the Oncology Research Professional, ORP, manual. The ORP manual can be accessed or downloaded from the SWOG website. Just click on the ORP manual link within the CRA workbench. Keep in mind there will be a lot of material to cover in a short period of time, so it's best to limit discussion to the material being covered. Once this 2-hour session is done, that will complete the clinical trials training course. Congratulations. Certificates of completion will be available at the registration desk following the session in exchange for your completed evaluation form. Evaluation forms are my favorite. That's how you can tell us what we can do better, maybe what we could cut, what we should add. We read through every word of it, so your cooperation with that is appreciated. Those attending virtually will also receive their certificate of completion once we review the participation records, and those will be forwarded by email, and you also will get a chance to fill out a survey evaluation form. So, before we get started, a few things to point out. Online data submission is required for all SWOG studies. Studies activated after August 1st, 2012 will be in metadata RAVE. Older studies still use the SWOG systems. Forms for these studies can be accessed by going to the CRA workbench on the SWOG website and clicking on RAVE Data Submission or Pre-RAVE Data Submission. Data submission requirements are found in the online version of the protocol on the CRA workbench and on CTSU. Only the most current versions of forms are online. Samples of current forms can be found in the master form set available on the CTSU website. With that said, I would like to introduce our first presenter, Larry Kay, Data Coordinator Extraordinaire. To know him is to love him. He's going to be walking you through eligibility, and as he makes his way up to the podium, please turn to page 33 in your manual. Oh, and I was prepared to reward the front row with Hershey Kisses, and I see you got a lot coming your way, you 2 lucky ladies.
Good afternoon. Thank you, Chris. Appreciate that. My name's Larry Kay, Clinical Research Data Coordinator on the Breast Committee and the Lung Committee. I'm not sure I'm an expert, but I'm a vet. So wish you a warm welcome to the Practicum. We're going to start out with a couple of exercises. I'll first give you a background information on a fall patient in a clinical trial, typical clinical therapeutic trial. If you turn to the page that says background information, patient ABC, last, first, middle initial, that will be the order of the initials. You are preparing to register patient ABC to a fictitious SWOG study, S2525, a breast cancer study. Usually SWOG studies begin with the letter S as in SWOG, FYI. If you see the letter E, it's probably another cooperative group study. Other patients at your institution are already enrolled on this study, S2525. S2525 is a one-step, phase 3 randomized clinical trial of drug X versus drug X plus drug Y for postmenopausal women with metastatic breast cancer. The objectives as stated in Section 1 of the protocol are the following. 1, to compare time to tumor progression in postmenopausal women with metastatic breast cancer treated with drug X versus drug X plus drug Y. 2, to assess the clinical benefit, CR, PR, or stable disease greater than 24 weeks. CR means complete response and PR means partial response, and overall survival for this cohort of patients. And finally, the third objective is to assess the adverse events of drug X as compared to drug X plus drug Y. Patient ABC was previously registered to a SWOG neoadjuvant treatment protocol when she was initially diagnosed with breast cancer. Neoadjuvant means before surgery. If she is eligible for S2525, she will be registered under her existing SWOG patient ID number, which is 123456. Okay, now some discussion points. Remember, it's important to understand the study objectives because it helps motivate compliance with data submission requirements. They're fairly explicit and they're usually located in Section 1 of the protocol. If a patient is registered to more than one SWOG trial, they will retain the same SWOG patient ID number. Otherwise, they are registered as a new patient. That's very important because there is an exception to that rule. It is for registration trials which have a unique patient ID number versus conventional SWOG trials. And finally, if the patient ID number is assigned by the Specimen Tracking System, or STS, when pre-registration specimen submission is required, you will use that same patient ID number to register the patient to the study and for all subsequent registrations to SWOG protocols. So some studies have a rare requirement to have a specimen submitted to a lab to enable the subsequent registration to that clinical trial. It will issue a SWOG patient ID number that will be the one that we'll use for all future trials on SWOG studies. Okay, let's see if this works. Okay, we're now to Section 5 of the protocol. Section 5.0 or 5 of the protocol is the eligibility criteria. On the page to your right, you'll see the Section 5 of the eligibility criteria. It's the slide on the right, or it's actually the page on the right, excuse me. This section may be used as a worksheet by the CRA to determine the eligibility of the study participant. This is not submitted data. It's just used as a worksheet. You don't need to submit the eligibility criteria as a general rule. This patient was registered to a prior SWOG study. Therefore, you will always use her SWOG patient ID number 123456, regardless of how many additional SWOG therapeutic trials she enters. Again, the exception is FDA registration trials such as S1605 and S1404. For patients enrolled to a registration trial, they will receive a new patient ID number for each study. The patient has met Section 5.1A and 5.1B eligibility criteria. Now, there are several tools used to help determine the number of days that separate two known dates. For example, the date wheel. Let's see if I have that. Does everyone have one of these? Please take one. It's a mechanical way of determining a date window for eligibility. There's also timeanddate.com. It's a hyperlink on the site, so you can access that and place the dates in to ensure that the patient meets eligibility. Exercise 1, we're going to discuss section 5.1c. So look at 5.1c in your handout. I'm not sure where it is, but it will say eligibility criteria at the top. Section 5.1c is towards the bottom. Take a quick glance at that. It's regarding measurable and non-measurable disease. You have a report of a CT scan done for measurable disease on 4 of October and a bone scan done for non-measurable disease on 17 of September. Let's complete section 5.1c and determine what was the last, emphasis last date the patient may have been registered in order to be eligible according to this criterion. If you have a date, we'll go ahead and try it out. That date is the 29th of October, 42 days after the bone scan and less than 28 days after the CT scan. I emphasize that try to be sure to schedule to accommodate both measurable and non-measurable disease, if it be the case for this patient or your patient that you're working up. Discussion point number 1. It is important to understand each time restriction in Section 5, since each eligibility criterion must be met exactly as written. No exceptions to Section 5 criteria are granted. Make sure to refer to Section 5, not the study calendar, usually Section 9, for baseline requirements. Again, Section 5, the eligibility criteria. If using a date wheel, please note that it may not have February 29th. So a day must be added in leap years. And finally, submission of the pathology report confirming the diagnosis is usually required with the initial form set. Remember to include the SWOG patient ID number, the SWOG study ID number, and the patient's initials on at least one sheet of the multi-page report, should it be multiple pages. Remove or redact the identifiers such as the patient's name, the MRN or medical records number, the social security number, address, etc. See Protocol Section 14 for data submission requirements and timing. Please also include all pages of the source document you are submitting, even if the final page is blank. Okay, on to exercise 2. Lab values are often required to determine the patient's eligibility and are necessary for assessing some toxicities. The following lab report was sent for patient ABC from CBC done on 20th of October for patient ABC. Exercise 2. Please determine the last date patient ABC may be registered according to Section 5.2a. So take a glance at Section 5.2a of the eligibility criteria, and you'll notice a window. That window is 14 days. So the last date would be November 3rd. Taking 4 of October date and the CT scan date, and the 17th of October date for the bone scan, the 17th of September date for the bone scan into consideration, if you tried to register her on the above date, would she be eligible? The answer is no. The last date available for this patient is still the 29th of October, so you must accommodate all 3 dates. Exercise 3. Reference Section 5.2a. If patient ABC had her labs done on the 14th of October, would she be eligible for registration on the 29th of October, according to Section 5.2a? The answer is no. It's 1 day off. It is 15 days between labs and registration date. Again, it's really important that you schedule these labs as accurately as you can, because otherwise it may cost the patient an extra lab to conduct. Discussion points. The timing of each criterion must be considered when determining eligibility. For example, in exercise 3, section 5.1c must be considered when determining the date of registration. It is vital that all lab tests and scans are done within the time specified in section 5. For example, in exercise 3, the labs must be done again for this patient to be eligible. Eligibility criteria are determined by the study design and objectives and are protocol specific. For example, the formula used here for calculated creatinine clearance is for female patients only. It includes multiply result by 0.85. Other protocols may use a more generic formula for calculating creatinine clearance. Your lab may report values in various ways. For example, 3.5 K microliters versus 3,500 microliters. However, it is very important to convert values into the format specified on the form. Formulas for units of measurement specified in the protocol must be used whether they are your institutional standard. Whether or not they are your institutional standard. So be aware, take a look at the values on the RAVE form and you'll see X, 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 for example, whole numbers, or X, X, point X, indicating a decimal. That should give you a good hint as to what the value should look like in the field. All questions regarding Section 5 of the protocol should be directed to the study data coordinator at the SWOG Statistical Center in Seattle, Washington. Okay, on to the on-study form. Now, in RAVE, the on-study form may have some sub-forms within that folder. We're going to discuss two of them, the lab values form and the patient and disease description form. First, the lab values. The on-study form is study-specific and collects data that the statistician will use in study analysis. It also collects data from Section 5 that will confirm whether the patient met the eligibility requirements set in the protocol at study enrollment or registration. Even if some of the information on the on-study form is provided elsewhere or repeated on other forms, it is important to complete all sections of the form. Furthermore, all SWOG study forms must be filled out completely so that the data is properly recorded in the database. In our example, you can see at the top the ANC, the hemoglobin, the platelets, the serum creatinine, and then the calculated creatinine clearance. For the record, the calculated creatinine clearance is derived. I'll discuss what derived means. It'll be based on values that are entered in the RAVE form. ANC is reported in this case as thousandths K per microliter in the report above. However, the on-study values form required this to be reported in cells per microliter. If 2.1 is entered as a value on the on-study lab values form, then the patient would be marked as ineligible. As you know, 2.1 can be converted or interpreted to 2,100. Also, please make sure to not include any commas when you are unabbreviating the appropriate lab test units on the form. For example, ANC is equal to 2.1 should be read as 2,100, not 2,100. Again, look at the the lab test units in the RAVE form, right where it says ANC on your example here on the page. And you can also take a look at the other example regarding platelet, or the platelet count. Notice it said 210 in the above example, and then in the rave form it says 210,000 or 210,000. Now on to the patient and disease description form that is on your right. You'll see two highlighted boxes. The patient and disease description form is unique to each study and will ask specific protocol-related data. It is important to complete this form in its entirety and accurately to ensure eligibility is properly assessed. Again, take a look at the form on the right side of the slide, and you'll see fields like performance status, height and weight, and then you'll see more boxes dealing with ER status, etc. This form indicates that all disease present at baseline must be reported. On-study forms collect sites of disease. We, the data coordinators, will verify consistency across all forms, including the baseline tumor assessment form and other uploaded reports. If you see the box below, you'll see some of the sites of disease indicated. Okay, on to the baseline tumor assessment, or BTA. This practicum deals with solid tumor assessment. For response criteria information on leukemia, lymphoma, and myeloma, please see the ORP manual in the SWOG-CRA workbench. The BTA documents all disease present at baseline. Identifying target lesions does not mean that other lesions can't be excluded. Exercise 4. First of all, take a look, if possible, read the two reports there regarding the radiology report. We're going to complete the baseline tumor assessment on the next page using the source document below. There's 3 paragraphs there. I'll go ahead and read all 3 reports. These are commonly called radiology reports or CT scan reports. Chest CT conducted on the 16th of October. There are extensive interstitial changes with a lesion in the right upper lung, now measuring 1.8 centimeters by 1.4 centimeters, and a parenchymal density on the right lung base measuring 2.5 centimeters by 1.4 centimeters. The impression? Lung metastases, or lung mets. CT abdomen pelvis, same date, 16th of October. A new low-density lesion is present in the right lobe of the liver, measuring 2.4 centimeters by 1.6 centimeters. A new hypervascular zone is present at the junction of the right and left liver lobe, measuring 2.6 centimeters by 2.3 centimeters. A new focal low density is present adjacent to the posterior margin of the right liver, measuring 0.9 centimeters by 0.5 centimeters. Impression, multiple liver mets. And then finally, there's a whole body bone scan conducted on the 14th of October. Foci of abnormal activity are noted in the skull, the left, 4th, and 5th ribs, and multiple foci in the thoracic and lumbar spine. Impression, multiple bony mets. There was also an MRI of the brain conducted on the same date, the 14th of October. It was negative for metastases. Discussion points. Okay, so you can see the scan reports. Using the above scans and the protocol section 10, please note that there are differences between measurable and non-measurable disease, section 10.1a and b, and target lesions versus non-target disease, section 10.2. There is also a difference between lymph nodes, and non-lymph node lesions under RECIST 1.1. Be sure to look at Section 10 because it will have the definitions of measurable and non-measurable disease and target and non-target disease. When completing the extent portion of the non-target disease, include as much information as possible. If lesion measurements aren't available, explain the degree of involvement of or extent of the disease process. For example, multiple, extensive, localized, etc. On older studies, bone scans are reported as radioisotope scans. Newer RAVE studies may have an option to select bone scan for assessment type. Bone mets are always going to be non-measurable disease, even if measurements are given by the radiologist. Again, refer to Section 10.1b. You'll see it explicitly stated. Documentation of negative tests can be important to note for some studies as an eligibility requirement. Please make sure to include all of the negative baseline reports/scans on the table of the BTA, shown on the next page as an example. If you can take a look at the page there on the right, you'll see a list of all negative tests. You'll see the test, MRI brain, and then you'll see the date. That's just a negative test. We'd like to know that. Lesion measurements should be documented in centimeters, which may require conversions if they are reported in millimeters. Again, please note the measurement. It could be 1 decimal point off and it makes a big difference. Always reference form instructions. The form instructions are at the top. You can see them on the right side here. You'll see RECIST 1.1. Right below, you'll see some form instructions there. Always reference form instructions. For instance, a max of 2 measurable lesions per organ and 5 lesions total. The sum of lesions field is derived once the form is saved, so please make sure all lesions are documented correctly. And finally, make sure the baseline tumor assessment, the BTA, is consistent and clean, since this can affect the formatting of follow-up tumor assessment forms, or FUTA forms, and lead to inconsistent data. If you need to make edits to the lesions followed, they need to be edited on the BTA first. So ensure accuracy by being consistent. Let's see, one thing I want to show. One more slide there. This is showing the extent on the baseline tumor assessment of the bone scan. Notice it says multiple. Okay, I believe that's it. Oh. Okay. Oh. Good luck to you all.
All right, can everybody hear me? Sounds like there's a little bit of an echo, maybe. Okay. Go on. Okay, I'm going to continue on. My name is Leah, and I'm a data coordinator at the Statistics and Data Management Center, and I am obviously presenting virtually, so the screen is very blurry for me, so I have the pages pulled up on a separate screen, but bear with me if I get a little out of order here. If we could advance to the next page, please. First, we're going to talk about protocol treatment. And for anybody watching virtually, this is page 41. So prior to completing a treatment form, make sure you always complete the vital status on treatment form if that's part of your study. This form updates the date of last contact with the patient And if it's not updated prior to entering the treatment form data, or honestly any other data, there's a high likelihood that a system query will generate. Completing the vital status form prior to any other data entry will help keep the data clean and avoid system queries for you. The specific information to be collected on a form is determined by the study design and endpoints, and treatment forms are very study specific. And the information documented on all forms is entered into study tables and is combined with the data for all participants in the study in order to conduct study analyses. Therefore, it's vital that all of the required or requested data is recorded on the form, and no field on any form is considered optional unless it is specified as an optional field. It's also extremely important that the data be reported in the format that it is requested. So looking at, any conversions you might need to make, and just looking at the type of data that's being requested. Keep an eye on that. And treatment forms will frequently ask for BSAs, which is a body surface area. And this is often used to determine the dose or doses of drugs that are given during treatment. And it's commonly referred to in protocols as meters squared. For example, if the dose is 175 milligrams per meter squared, you would multiply that BSA number by 175 to get the correct dose in milligrams. And we do have a BSA calculator on the SWOG website in the CRA workbench, which is found on SWOG.org. And it's in the tools of the trade link, where there's also a bunch of other handy tools and other calculators, including that BSA calculator. So be sure to also recalculate that BSA if the patient has had a weight loss or a weight gain of greater than or equal to 10%, and always use the actual body weight unless otherwise specified in the protocol. If the BSA is requested on the treatment form, you must enter the information on every treatment form you submit, even if it hasn't changed. All right, so our first exercise, we're going to be looking at our patient ABC again, who was randomized to arm 2 of a study. And the dosing instructions are in protocol section 7. And we're going to look at completing some fields in a treatment form using the information that's below. And we're first going to look at the treatment table, which is kind of in the middle of the screen there, where it says arm 2, drug X plus drug Y. So for drug S, it's an intravenous drug, and the dose is 200 milligrams per meter squared. So that's 200 milligrams times the BSA. It's received on day 1 of the cycle, and the cycles are 28 days long. Patient is also going to receive drug Y, which is an oral drug taken twice a day, and each pill is 50 milligrams. So they receive a total of 100 milligrams per day, and it's taken on days 1 through 21. And the patient will keep a pill diary, and that pill diary and bottles are returned to monitor compliance. All right, and then if we look, I just saw in the chat, am I on the wrong page? My page says 42, but I'm sorry if I led you astray. But we're on exercise 5. So looking kind of back up at the top of the page, where we have a little excerpt from some clinic notes for patient ABC. As they return for cycle 2 on December 1st, toxicities were assessed, and there were no dose reductions needed for cycle 2. And then labs were done prior to the infusion of drug X. And the patient was given 352 milligrams of that intravenous drug X, over 30 minutes, as indicated in the protocol. And she was given a new bottle of the oral drug Y. And the patient returned the cycle 2 bottle. Okay, I'm reading that we are actually on page 46. I'm sorry. Okay, so patient returned the cycle 2 bottle and the pill diary. And it looks like she missed the last 3 days of oral drug because there were 6 pills left, and the patient takes 2 pills per day. So that's just something to note. And then the patient also experienced grade 2 nausea after infusion of drug X that was treated with antiemetic medication. All right, so we're going to come back to these discussion points, but if we could advance to the next page. All right, so this is what a form could look like, a treatment form could look like while you're filling it out, while you're completing it. And we're first going to look at that first purple box outlined that says date of last treatment. And of note, this should always be, as it says, the date that the last treatment was received. In this case, or this would be the last date that oral drug was given or taken. And, oops, sorry, could we go back? There we go, okay. Forward, there. Okay, okay, so for the date of last treatment is the date that oral drug was last taken, which given the treatment start date and the patient taking treatment for 21 days, you might expect that to be December 21st. But since they missed the last 3 days of drug Y, the last date they received that they took that drug was December 18th. And note that that also doesn't match the reporting period end date that's kind of above and to the left, because the reporting periods or the cycle lengths are 28 days, but the treatment start date and date of last treatment can be a maximum of 21 days because they only take the oral drug Y for 21 days. All right, and then you see below that, we did have an unplanned dose modification, which we'll talk about in the next purple outlined box, the treatment table. And you can see for drug X, the intravenous drug, our dose planned is going to need to be the dose given before it's multiplied by the BSA. So this is what would be indicated in section 7 of the protocol. And you can see that above the table, there's kind of a key there to tell you what to enter. And it says for drug X, report planned and delivered doses in milligrams per meter squared. And then the total dose is going to be the total milligrams given. So our dose planned and our dose delivered for drugs X are 200 because there is no dose modification for this drug. And that times the BSA, which if you look a little further up, our BSA is 1.76, comes out to a total of 352 milligrams. And then for modifications, there weren't any dose modifications for drug X. So if you look at the key at the bottom of the page, we have a 1 entered, which correlates to no dose modification. And then looking at drug Y, we did have a dose modification, but it wasn't planned. It happened during the cycle. So it was planned for the patient to receive 100 milligrams per day. And you can see, I forgot to mention, above the treatment table for drug Y, you're going to report the planned and delivered doses in milligrams per day. And so we have 100 milligrams per day was planned. And then the dose delivered, this is where there might be some room for where you could argue one way or the other. The patient didn't receive the full 21 days of drug. And so we have 50 entered here, but you could also probably just enter 100 because they did receive 100 milligrams of drug for the first 18 days. Anytime you're not sure what to enter on something, I would recommend either reaching out to your data coordinator or leaving a note in the comments section of the form explaining why you entered something a certain way. And that's just a good way to avoid queries. And then looking at the total dose given for drug Y, since it's they receive 100 milligrams per day, and they took 18 days of drug. That comes out to 1,800 milligrams. And for a modification, since the patient missed a few days of drug, we have number 10, which you can see correlates to dose missed. All right, so can we go back one page, please? Forward one. Perfect, right there. So we're just going to go through the discussion points. You want to ensure that in a real world situation, the vital status on treatment form is completed. Just stating that again, because it's very important. Otherwise, you will get queries if you don't complete that before entering data on the treatment form. And then as we discussed, the reporting period start date and end date may not be the same as the treatment start date and date of last treatment. Like in this case, the patient receives protocol treatment for 21 days, but these cycles are 28 days long. And those reporting periods reflect the length of a cycle, including AE assessments. So AEs are assessed for all 28 days, even though the patient's only taking the study drug for 21 days of the cycle. And treatment dates reflect actual treatment given during the cycle. Reporting period end date should always be day 1 of the next cycle. And that usually will be indicated in the form instructions. So for this one, December 29th was the last day, was the reporting period end date, and that should correlate to the next cycle's day 1. For weight dependent doses, it is important to weigh the patient prior to each infusion, so on that first day of the cycle, so that the correct amount of drug is administered, as referenced in protocol section 7. And generally, when a DC is reviewing study data, the dose the patient should have received will be calculated per the protocol. But if there is a discrepancy of plus or minus 10%, the institution will be queried if there's no explanation given on the form. So that's where that comment section of the form comes in handy if you need to explain anything, or email us. And please always refer to the protocol contact information and section 7 and 8 of the protocol for study chair contact information, because that's who you should be contacting about dose modifications or any kind of treatment questions. All right, can we advance to the next page? Okay. All right, now we're going to talk about dose modifications. And these are usually talked about in section, or they are talked about in section 8 of the protocol, which specifies the toxicity criteria to use, which will just be which version of the CTCAE. And it defines anticipated toxicities and dose modifications. So let's look at, in this scenario for our patient ABC, what kind of dose modification they could receive for drug X, which is our intravenous drug. So looking at that table, are we on the right page? Go back one. Oh yes, okay. We're on the page on the right. I don't know what page number it is because my page numbers aren't correct, but we're on exercise 6. So looking at that dose modification table near the top of the page, for drug X, the IV drug, which the full dose would be 200 milligrams per meter squared, but they can decrease that by 1 level to 100 milligrams per meter squared. And that's if there is AE toxicity grade of 3. If it's 0 to 2, okay, somebody just said it's page 48. Thank you, Vicky. So if the AEs are all 0 to 2 in grade, there's no change needed. The patient can receive the full dose of drug X. If there's a grade 3 AE, you would decrease it 1 dose level. So they would receive 100 milligrams per meter squared rather than 200. And then if there's any AEs of grade 4, you would discontinue the drug altogether. Okay, so as previously stated, our patient was randomized to arm 2, which means they received that drug X on day 1 at 200 milligrams per meter squared. And prior to cycle 3, she had grade 3 ANC adverse event. And so looking at that table in the BSA on the opposite form, which we'll look at in a second, we're going to determine what the dose for cycle 3 should be. And just of note, our oral drug Y, didn't require any kind of dose modification. So they received the full dose of that. And so can we look at the next page, please? Okay, here's another look. Here's a look at how the treatment form would look in RAVE, our EDC. And looking at that first kind of teal outlined box, we're saying, yes, there was a dose modification and it was planned because this patient had that ANC determined before cycle 3 drug was started. So we're just going to look at how to report that in the treatment table in the middle of the form. So for drug X, instead of receiving the full dose of 200, she's going to receive 100 because she had the grade 3 ANC AE. And so that dose delivered is also going to be 100. And since the BSA was 1.76, we're going to times that by 100 and get a total dose of 176 milligrams. And then looking at our modifications, we selected dose reduced, which might show up as that number, which you would just refer to the key below. And then in this case, it also asked for the reason, the dose modification reason, and it was an AE. So once you save and submit that form, it will probably generate another form, which is kind of shown on the bottom half of this page, the dose modification form. And this is asking why drug X why there was a dose modification. And we have 1 selected, which correlates in the key below to an adverse event. It asks for the number of days delayed, but the dose wasn't delayed, it was just reduced. So you would leave that blank. You don't need to enter anything in there. And then it's asking for the type of AE, which for ANC, it correlates to a blood and bone marrow type of category, which correlates to 2 on the AE code at the bottom of the page there. If we could go back 1 page, we'll go over the discussion points. Okay. So as we just discussed, this dose modification required the treatment for cycle 3 to be dose reduced by 1 level to 100 milligrams due to the grade 3 toxicity. And you'll note complete the modification fields below were answered yes. Wait. And then such instruction must be followed or a query will be sent requesting the information. And this can be done on the dose modification and dose modification reason, that second form we just discussed. And remember to report the actual doses given according to your institutional treatment records, because any discrepancy between what's in the records and what's reported we'd want to have that explained in the comment section. So like, keep drilling that in. The comment section is your friend. And so are the DCs if you want to contact them. All right, can we advance to the next page? There we go. I think, is that baseline abnormalities? Yes. All right, so now we're going to look at the baseline abnormalities form, which some studies use to document existing conditions or continuing toxicities caused by prior treatments. And this is all prior to being enrolled in the study. It says corresponding sections from the CTC are included in handouts, but I don't think we actually had those handouts. just one of the setbacks of presenting something virtually as we are now. But your institution should have some kind of resource for the CTCAE that you can always refer to, that will be probably referred to quite often. But let's look at an exercise for AEs, for baseline AEs. So this is our same patient that had a physical examination prior to being enrolled in the study. They're recovering from a cold and presents with a mild cough with only non-narcotic medication. And she's been experiencing some shortness of breath, mild fatigue relieved by rest, mild difficulty sleeping, and mild headaches that do not affect function. Oh, are we on the correct page? Yes, we are. Okay. And which condition cannot be graded without further information? We have this pre-filled. As I stated, we presenting virtually. This is how we have to do it. But the condition we can't grade without further information is dyspnea, which refers to the shortness of breath. And the information we would need for that is the level of exertion that causes the shortness of breath. If you look at the CTCAE dyspnea AE table, that's how you determine how to grade it, is what level of exertion was causing the shortness of breath. And where would you get this information if you don't have that in your clinic notes? And it would be from the treating physician or the patient. I guess, can we look at the next page? Oh wait, it's already up, never mind, stay there. So if you look at the page on the right, this is how it would look if you were completing this in RAVE for the baseline abnormalities. And as you can see, the way the other AEs were described in those clinic notes, we just know that they are going to be a grade 1 because there was enough information for us to grade them. And you can see the dyspnea has unknown entered. You can't actually enter unknown. So you would just have to, I would just recommend not entering that until you have the information to report. Yeah, so that's why grade 1 is selected for those other ones. So, all right, and then looking back at our discussion points, not all studies require a baseline abnormalities or medical history form, but always refer to section 14 of your protocol to determine which forms are needed. And the clinic notes may not always use the correct terminology to describe an adverse event. For in this example, we had shortness of breath, which refers to dyspnea, or they might say difficulty sleeping, which would have its own term. And to find the correct terminology, there's a link to the CTAC terms on our CRA workbench under tools of the trade, or as I said, you might have this resource at your institution. And please use as specific a CAE terms as possible, the best as possible, instead of the other category. Just ensure that you've done the best you can to select a term out of the options available before selecting other. But when you do have to use generic terms such as pain, make sure you specify the site or type of pain in the comment section at the bottom of the form, and consult with the PI if you have any questions regarding a certain AE term. And the baseline abnormalities form is not used as a record of the patient's full medical history. For example, a prior tonsillectomy or ongoing controlled diabetes should not be recorded, but it is used to record current conditions and existing toxicities only. For example, fever or hypocalcemia should. And then in terms of a medical history form, this is a way to report the complete medical history prior to registration, and it encompasses all prior conditions and procedures. And that's how it kind of differs from a baseline abnormalities form. All right, can we advance to the next page? And now we're going to discuss reporting adverse events. And the adverse event forms are used to assess and document toxicities to monitor the effects of treatments. Nearly all protocols require AEs to be assessed within a certain window of the next cycle of treatment beginning. When completing the adverse event assessment form in RAVE, please be sure to use the most recent date of AE evaluation to ensure all AEs from a cycle are captured. So that's going to be the date, the last date the AEs were assessed before the next cycle of treatment was started. And in exercise number 8, we're going to use the information from the clinic notes below, and we would use excerpts from the CTCAE, but we don't have that on hand right now. But we're going to complete the cycle 1 AE form, as you can see on that opposite page. And in our clinic note for our patient ABC, who we completed that baseline abnormalities form for, patient ABC continues to experience headaches, insomnia, and fatigue. And they've all increased to grade 2, but are not related to treatment. And during cycle 1, she had some loss of appetite and nausea without alteration of eating habits during the first few days of treatment. And she had 1 episode of vomiting on day 1, which didn't require IV fluids, and all are definitely treatment related. And today, patient presented with mild ankle edema, less than 10% changing in circumference, and lab results showed white blood cell decreased 2.5 to 2.5K by nanoliters, both probably related to treatment. And she will start cycle 2 tomorrow. So do we have enough information to grade each toxicity? I'm going to tell you that yes, we do. And yes, we can assign attribution and status codes to each, as you can see on that opposite page. Based on how they were described, we're able to assign a grade to each AE. And if you want to look a little bit more into this, you can check out our CTCAE document on the CRA workbench to see how each of those are graded. It's a good exercise. And then it was specifically said in the clinic notes what the attribution for each one was. So that makes it easy to assign an attribution code. All right, I guess we'll go back up to our discussion points. Not all protocols will include status codes on the AE forms. It depends on the type of study, how much information will be asked of each adverse event. So it's good to find that out ahead of time. We always want to refer to the instructions on the AE form, because some studies may only want certain grades reported, while others may include or exclude baseline conditions. And most protocols require attribution codes, which are assigned only by the treating physician. So if you need an attribution, if the clinic notes you have on hand are missing an attribution, you need to refer to the treating physician for that information. And in the CTCAE, many general terms such as edema must now include the site of toxicity, such as in this case, the patient had ankle edema, so that's specific enough in this case. And this form includes edema in her limbs, but if the patient had facial edema or edema in her trunk, those sites would have to be documented, specified, and graded separately. So if the patient had edema other than just in the ankle, those would be reported as 2 separate AEs and followed separately as well. Note that the adverse event start and end dates will not always be the same as the start and stop dates of treatment, but should match the treatment reporting period dates. So those are the cycle dates, which, as we discussed, may not match the dates in which the patient received treatment. AEs should be assessed at the end of the cycle, ideally on day 1 of the next cycle prior to treatment. Please make sure the reporting period end date is also day 1 of the next cycle to avoid any system queries on the form. Okay, and with that, I'm going to pass it off to Chris to discuss the follow-up tumor assessment forms. Thanks for bearing with me, everyone, as we presented virtually, and I hope you all have a wonderful stay in Seattle. for those of you attending in person.
Okay, so actually she's passing it off to Tracy. Chris will do our final
Sorry, Tracy.
section. That's okay. It'll be on your next, no, I'm kidding. Thank you, Leah, that was great. Okay, so first of all, we've been sitting here for a while, and I'm going to borrow a term from baseball, and without necessarily leaving the room, do you want to just stand up and move around for a second? I mean, you know, let's keep the blood flowing a little. And I will talk to you in the meantime about how amazingly clear those clinic notes are. I mean, your notes in your hospitals always look like that, right? I mean, it's got the attribution right there. It's got, oh, and especially like the CT scan reports that have the highlighted, like really bolded, you know, this is the, yeah, right. We know. All of this is fabricated for the ease of doing these exercises in this type of format. So we know it's all fiction, but we're trying to make it as seamless as possible. Okay, so now, if you mind sitting down again, and we will continue on. So I'm going to be talking today about response assessment. Response assessment, I'm going to take my mask off. I'm so used to this thing now. Oh my God, my face. Okay, response assessment is a really important part of especially phase 2 trials where we're trying to figure out if a treatment is effective or not. So accurately recording your tumor measurements is really important, and being able to follow those tumor measurements throughout the course of the study is also really important. So make friends with your radiologist, because otherwise it can be difficult. Okay. So a patient's disease response to therapy is the primary endpoint, as I said, of most phase 2 studies, and the secondary endpoint of phase 3 studies. To assess a patient's response to therapy correctly, the study chair must have accurate tumor assessment on disease sites. Phase 2 studies are particularly sensitive to errors in recording response factors. Incorrect response assessment may result in needless exposure of patients to an ineffective agent, or early study closure, or incorrectly labeling an agent as ineffective when it might be, or a biased response rate estimate. So the only thing we're going to talk about today really is the RECIST criteria. RECIST is the response evaluation criteria for solid tumors. Solid tumors are those that can be palpated or measured using radiography. They're the types of things that you see with lung cancer, breast cancer, GU, GI, but do not extend to hematologic cancers like myeloma and lymphoma. And, well, lymphoma is kind of in a class by itself. But myeloma and leukemia are considered hematologic cancers, and those are mostly assessed using lab values. And I think Larry might have mentioned earlier that if you're doing assessment in leukemia or myeloma or lymphoma, there are guidelines to how to do that response in the ORP manual on the CRA workbench. But the main thing to remember is that you should be looking at section 10 of the protocol that your patient is on. Every section 10 has, well, I would say every, but some section 10s have some variation in the RECIST criteria. They'll have a special rule, or they'll have a guideline on what type of assessment can't be used or something. But for the most part, what we have in front of you here is just a very generic version of section 10 for a measurable, for a solid tumor study. And there are some basic guidelines for doing response assessment that we've been promoting for many years. For one thing, always use the response criteria and schedule given in the section 10 of the protocol that your patient is on, as I just said. Every disease assessment called for on the study calendar in section 9 must be done on time, or as close to as possible to on time as you can. All disease present at baseline must be documented to ensure that response can be properly assessed. Each site of disease must be assessed at every scheduled disease assessment unless otherwise specified. For some sites, like I know for lymphoma, if they do a bone marrow draw to check for marrow involvement at baseline, they usually don't have you do that at the next assessment, which might just be CT scans. So something like that would be specified in the protocol. The same method of measuring the disease used at baseline must be used at each assessment. If you're starting off with an MRI scan, really keep that in mind, because insurance companies may not pay for an MRI scan every 3 months. Usually a confirmation assessment is required after an initial observation of response, and it should be scheduled according to the protocol instructions. A lot of protocols will say schedule a confirmation response within 4 weeks, or after 4 weeks after the initial response is noted. So that should be also outlined in your section 10. So now the section 10 that I have here does have some definitions of measurable lesions, like we talked about earlier. If you're using CT scans, it's usually 1 centimeter. If you're using palpation, you can have a 2 centimeter lesion would be considered measurable. But the other thing I want to talk about is target lesions and non-target lesions. So target lesions have to be measurable. They also are, if you have a lot of lesions, if your patient has a lot of lesions, we're only going to select 5 of those lesions to be target lesions. These are RECIST rules, not our rules, but RECIST. And if you have several lesions in one organ or in a paired organ, like if you've got kidney lesions and you've got 2 on the left side and 1 on the right side, you're still only going to follow 2 of those lesions. So it's 2 lesions per organ for your target lesions. You still want to follow all disease that was present at baseline, but your target lesions are going to be limited to 5 lesions and only 2 per organ that's involved. So pick out your best ones, I guess. I don't know. Your radiography, I know a lot of radiologists are clued in to RECIST and will just say, okay, this is the lesion that's going to be best to follow. So take your cue from them, I guess. Another thing that we talk about in section 10, SWOG always has what we consider a best response. So best response would be, like say your patient has stable disease after a certain amount of treatment, or maybe they've had a PR. They've had a PR, that's great. And then a month later, they've had a progression. So, you know, the final outcome is progression, but there was something good that happened there. So SWOG, we always try to find the positive in these things, and we're looking for the best response. So we'll be talking about best response in the next few pages. And you're probably familiar with most of our definitions of response. Complete response would be complete disappearance of all lesions. Partial response is a 30% decrease in the sum of your measurable lesions. We're not looking at individual lesions, but the sum of all of your lesions has to decrease by that amount. Stable disease is, okay, they haven't increased enough to be progression, but they haven't decreased enough to be a PR, so they're kind of in that stable zone. And then progression would be a 20% increase. So let's move on. Yeah. Okay, so let's talk about our patient who has been receiving treatment on S2525. Okay, so at week 12, after 3 cycles of treatment, we're repeating our CT scans for this patient. And what we have now is, let's see, a chest CT that was done on January 20th, which is showing extensive interstitial changes with the lesion in the right upper lung, which now measures 2 centimeters by 1.6, and parenchymal density in the right lung base measuring 2.5 by 1.5. The CT of the abdomen and pelvis shows the lesion in the right lobe of the liver now measures 2.5 by 1.5. The hypervascular zone at the junction of the right and left liver measures 2.5 by 2.5, and focal low density adjacent to the posterior margin of the right liver measures 1.5 by 1.5. So our measurable lesions haven't changed a lot. There's been a little shift here and there in size, but we very conveniently captured the baseline tumor assessment measurements here, so you can refresh your mind on what those are. And you'll notice that at baseline, we had a sum of lesions that was 9.3. And after we've totaled up, filled in our form here, first off, when you start completing a follow-up tumor assessment form, the first section that pops up is, was their disease assessment done? Yes. Were there any new lesions? I mean, it's going to ask for the assessment date also. But then, were there any new lesions? And in this case, no, there were no new lesions, so that's blank. We just leave it blank. And it says there is a question down below, was there any symptomatic deterioration also? Now that symptomatic deterioration is another form of progression, and I used to see this from time to time in GI patients where the lesions may not have changed, or they may not have even been on study long enough to have as follow-up CT scans, but they're unable to continue with treatment because of, say, an inability to swallow or something. So they're definitely progressing, but not necessarily based on the size of the lesions, but just their general overall condition. Okay, and then the next question, of course, is has this patient progressed or relapsed per the definition in section 10? Well, no, they haven't. So our target lesions, we put our measurements in here according to the CT scan, and you see now that our sum of lesions is only changed by 9.5, 0.2. So this patient definitely is still stable at this point. Another thing I like to do when I'm working on measurable lesions of this sort is I sort of sit down and figure out what would a 20% increase be? What would a 30% decrease be? And kind of have that somewhere in my notes so that I know that if this patient gets to 11.2, for instance, that's definitely a 30%, 20% increase, and this patient will have had a progression at that point. And then for our non-target lesions, the extent on the RAVE form, there are only a few options that you can choose from on the RAVE form, so you're pretty limited in that. It's, I think it's present or, let me see, where are they? Oh, complete disappearance, present, unequivocal progression, or not assessed. So somewhere in that realm. If your physician says that there's been a definite progression, then we usually ask that you describe a little bit about how that was determined, since we're not actually getting that much CT information on, like we do with the target lesions. Especially if it's something like a pleural effusion or some type of non-measurable disease, it's like, well, I think we kind of need to know how that was assessed. Okay. All right, I'm missing a page. Yeah. Okay, I'm missing a page, just in my notes. Okay. So at our week 24 assessment, just out of order. Found it. At our week 24 assessment, the patient's CT scan is showing that there continues to be extensive interstitial changes within the lesion in the right upper lung, which is now measuring 2 centimeters by 1.6, and a parenchymal density in the right lung base measuring 4.5 by 2.5. The low-density lesion in the right lobe of the liver is now measuring 3.5 by 3. The hypervascular zone at the junction of the right and left liver lobe is measuring 3 by 2.5. Focal low density adjacent to the posterior margin of the right liver is measuring 2 by 1.5. So things are not looking so good for this person. And the abdomen and pelvis is showing new hypervascular solid masses scattered throughout the liver, the largest being 5 millimeters. The impression is increased liver mets with new sites of malignancy. So things are not looking too good for this person. For one thing, any new lesions that come up with RECIST, that is pretty much automatically a progression right there. If there's a new metastatic lesion that was not there at baseline, then that's a problem. Another thing that happens from time to time is there might have been a suspicious lesion at baseline that is suddenly looking worse, and your physician sort of says, you know, maybe that is a metastatic lesion. Well, you can't just add that at this point to a follow-up tumor assessment form if it was there at baseline. What you can do, though, is add it, go back and amend your baseline tumor assessment form and add it to any other follow-up tumor assessment forms that you've done in the meantime and update that lesion on all of them. So you do need to add them, and this is something that you'll get a query about, probably. It's like if you don't say that, or if you suddenly have a new lesion, but you're saying, oh no, it was there at baseline. It's not a new lesion, really, it was there. We're gonna ask you to go back and add that to your previous forms. Okay. So they also did a whole body bone scan, which shows foci of activity are again noted in the skull and the left and fourth and fifth ribs, which were there at baseline. But there is increased uptake in the thoracic and lumbar spine. So the impression is increased uptake in existing bony metastasis. Now, there's a little note buried in your section 10 on RECIST criteria that says that if the only thing that you're going by is a body scan, a bone scan like this, that in and of itself is not indicative of progressive disease. If the rest of the patient's clinical picture is stable, but there's increased uptake on the bone scan, you really need to have another type of assessment done to say that the patient has actually progressed. Bone scans can be affected by a lot of different things, degenerative joint disease, arthritis, that can show also uptake, increased uptake on a bone scan. Okay, so at this point, our sum of lesions, now that we've plugged them into our form, is showing 13 centimeters for total. So we know that this patient has progressed, and we're going to also note our new lesion on the first part of the follow-up tumor assessment form. Yes, there were new lesions according to the CT scan in the liver, and we've entered all of our lesions, and we get the 13, and then on our, okay, for our non-target lesions, we're showing unequivocal progression. Now this would be based on the clinic notes, based on your clinician's assessment. And in this case, looking at the entire clinical picture, he's saying that there's unequivocal progression on both the CT scan and the bone scan. And I think we can let it slide in this case that this patient is definitely progressing. So that would be your outcome there. Now, for most protocols, that would be a reason to take the patient off treatment because we're not going to, there might be another more effective treatment for your patient. So let's get them off this one and onto something better. The next page is just a really brief summary of some examples of when response assessment didn't quite go that well. So, or wasn't documented as well as it would be nice to have. We have measurable lesions at baseline, and at week 6, it looks like the patient has had a PR, but it's an unconfirmed PR. You're supposed to schedule a confirmation assessment. And at week 10, one of the lesions was on the radiograph, it was on the CT scans, and the other one was not. And all we have there is a note that says measurement not noted. So it's like, okay, well, do you talk to your radiologist about that? Talk to your physician about that? Try to get a measurement? As it is, if this was the only information we have, that would just be an inadequate assessment. The next exercise is pretty similar. Measurable lesions at baseline based on CT. And again, at week 6, there's been a PR. And then at week 12, zero disease. Zero disease. It's a CR. Well, no, it really isn't because that was an X-ray and the other lesions were assessed by CT scan. So it really has to be the same assessment method every time. We can't compare X-rays with CTs. And our final one is a measurable lesion by palpation and one by CT and one on a bone scan. And we've got one objective status of stable, one of PR, and then it looks like the measurable lesions are completely gone. 0 by 0, 0 by 0, it's great. But remember that bone scan is still positive. So that means this patient still has disease. The best we can do in this case is a PR. So that is about it for response assessment. And I think we are getting close to the end of this. Okay. Christine is going to come up and talk to you about follow-up forms and how to record things that happen after the patient has finished their protocol treatment. Thanks.
Hi. I never introduced myself. My name is Christine Magner. I am a data coordinator at the SWOG SDMC. I work on GI and rare tumors. I come to you with some good news. We're almost done, and we're going to end early, so congratulations on that. I want to use a little bit of that bonus time to put on my SWOG cape and defend our department just a teensy bit for 1 minute. If this is your first practicum, you really don't know any different. If this is not, you're probably thinking, what is this? So let me just really quickly explain to you. We're in uncharted territories here. We have never done a hybrid meeting. I realize you've probably never attended a hybrid meeting. Last year we did it fully remote, and sorry to break away from script, I'll get off my soapbox soon. We shortened the practicum from about 3.5+ hours to 2 hours, and today it's going to be even less. You're welcome. But we've had to take away probably half the content. And so normally we would work through exercises and we'd be at a table and there would not be a pandemic and we would all have handouts and we'd go through these together and we'd work interactively and we'd ask questions and we'd answer them and we'd come off really smart and you'd be so glad to be here. And now we are standing at a podium talking to some of you in the room and some of you online. We don't have anything to point at and look at and share, and we can't really work through things. So I know it's been clunky, and I don't want this to be your first impression of SWOG. We're better than this, and I say that at a risk, and I understand that. please have faith in SWOG. We know what we're doing. If you want the full practicum experience, you can experience that online anytime you want at SWOG.org. I encourage you to take a look at it. If you have any questions, we're still monitoring chat. We still have microphones. If anything is unclear, yeah, we know. We tried. We tried real hard to make this kind of smooth, and it's rough. So thank you for showing us grace. Thank you for being patient with us. Again, we're happy to answer any questions. I'm going to close this up by talking about follow-up forms and notice of death forms, which is typically how you're going to end your patient on a protocol. We had practices to work through to explain it all, and again, in the interest of time, we've taken those exercises out. So essentially, I'm going to show you some forms, and some of you can see them, and some of you can't, and that's just another great part of today's attempt. Again, we thank you for your grace and your patience. Oh, yay, thank you. Thanks to Larry and Leah, who really had it rough being remote, and Tracy and Larry, so thank you to them. Thank you for your patience. This is always a really long day with so much information thrown at you. So if you get back to your sites and get your thoughts straight and have questions, always reach out to our office. I think we've shown the distribution list 150 times, and it'll be 151 by the time I'm over. So, thank you for the sidebar. Let's talk about your patient that has now progressed and they're going to come off study. SWOG protocols used to be very standard. If your patient progressed, they come off study. And that's no longer true with some protocols. They can progress and stay on study. So we're going to talk in general SWOG terms, which is what this training course is all about, just general SWOG terms. There's always going to be exceptions, especially with FDA trials, but even more so with standard SWOG trials now, which they're not really standard anymore. So, your patient has progressed and they're going to come off study. Forgive me for not making eye contact, but because I also can't read that screen and I may or may not be ready for readers, so I'm going to look at this book below me. So, as you determined when completing the FUTA for patient ABC, she has progressed and protocol treatment will be stopped. The off-treatment notice must be submitted with the FUTA form. It's imperative that every section of the off-treatment notice is filled out. That includes reason for going off treatment, planned post-protocol treatment, etc. As always, document any unusual or additional information in the comment section. Almost every single RAVE form has a comment section, and that is by design. You can explain a lot away in the comment section. If ever you're putting something that just doesn't look right, or you know is going to make us say, "Can you explain this?" Explain away in the comment section. That will avoid a query. We'll accept that form usually. Good to go. So use the comment section. There's a huge character limit on it, so explain it away. So you are seeing what the off-treatment notice looks like on your screen. Again, we've talked about the vital status forms, both on treatment and off treatment. Going forward, all studies are going to be using those forms, but some of the older studies have not transferred over to them. So if you have a vital status form, you would complete the one for off treatment. This means you're done completing the on-treatment vital status forms and you'll only be completing the off-treatment vital status forms. If not, you will have the fields on the form displayed here, which is vital status and date of last contact. So, off treatment. Did the patient receive any protocol treatment? Most of the time, this is a yes, and we get an overwhelming amount that say no, and I get a little snarky sometimes and say, you state that patient did not receive any protocol treatment, yet you've submitted 14 treatment forms. Please verify your entry. I don't get snarky often, but when I do, I go for it. So we're just asking, overall, has the patient received any protocol treatment? We try not to do any trick questions on these. We want to be as straightforward as possible. So the off-treatment reason, you do have designated options. Either the treatment is complete, which is only an option on select SWOG studies, adverse events, patient withdrawal, disease progression, death, other. You'll notice that a few of those have specify with them. If you select that answer, you will need to specify. Being specific, obviously, when you specify, would be appreciated. If you give really generic stuff, we're probably going to query you because this is where we're getting our information. I will say another big hiccup we get is a lot of off-treatment notices that state the patient is off due to progression, yet we have zero documentation of progression. So when we receive these, a common query is you state patient's off due to progression, but you've not noted any progression, which means we're expecting you to submit a FUTA form and document that progression. Speaking of which, if your DC gives you a query that doesn't make sense to you, I make a lot of mistakes. I'm going to let you just be honest, because I swear I looked at something, but then maybe I got interrupted and I'm looking at a different chart. I mean, it happens. I mean, none of us are perfect. If you think we're not being clear with our query or we've queried you wrong, you're probably right. Give us a call. We'll be like, well, this is embarrassing, and we'll delete it. So never, I'm a big advocate for reaching out to your data coordinator. So in this case, your patient is off due to progression. You don't need to specify any further for the specify boxes except for the one that matches that. In this case, we're going to be asking about the progression. So you're going to have to give some information. If you just put progression under specify, depends on the day the DC's having, you might get a query. Okay, yeah, I get they progressed, but could you tell us where and how? So. And then we're looking for the off-treatment date. The only tricky thing here is that if your patient is off due to progression, we're looking for the date of progression, and that is the scan or test that documents that progression. Not the day that it was read or the day that somebody talked about it. It's the actual date of the scan. We've been told no longer to query for that, but we're secretly thinking it. So date of progression equals whatever the documentation is of that progression. If it was a decision to be made, not for this patient because it was progression, then that's totally acceptable. It doesn't have to match the last treatment date. It is the actual date that the decision was made to discontinue treatment, or the date of death, or the date of progression. Will the patient receive any further treatment? We'd like to know. Sometimes you don't know, so unknown is an option. If you say yes, we do request that you provide the regimen name. Start dates would be great too. And then again, the comment section. Anything you might want to share, feel free to do that. Always refer to Section 7 for criteria for removal from protocol treatment and Section 14 of the protocol when completing an off-treatment notice, as other documentation is usually required. When submitting an off-treatment notice, make sure that all treatment data, toxicity assessments, and FUTAs are also submitted. End-of-treatment specimens may be required to be submitted. Refer to Section 15 of the protocol for further clarification. So now we've got again, the off-treatment vital status form, and lots of things to discuss here. For studies that have the vital status form, this form must be completed prior to completing the follow-up form. The date of last contact is derived from the vital status form and cannot be updated any other way than by completing the vital status form. And if any of you have made that mistake, it's a tough lesson, but boy, do you learn quick, because it's a real pain. And then you got to contact us, and then you got to wait for us to call you back. And start with the vital status forms first. It will save you headaches, I promise. Once a patient is off protocol treatment, a follow-up form is required at each follow-up at the protocol specified intervals, at first progression, and at time of diagnosis of second primary. If the study uses a vital status form, please update the status and date of last contact prior to completing any other forms in RAVE. All fields must be completed. Clinical assessment cannot be done by phone. In this case, the progression was previously reported when patient was removed from protocol treatment. Therefore, the notice of first progression question is answered as no. Again, we're looking for previously unreported. It was already reported, and that's why the patient came off study, so there's no need to report it here. A follow-up form is due, outlined in Section 14.4 of your protocol. It could be 12 weeks, 3 months, whatever it is. We don't need one when the patient comes off study. The off-treatment notice is essentially what kicks off your follow-up forms. When documenting progression on the follow-up form, be as specific as possible in reporting the sites of disease. If patient went off treatment for reasons other than progression, FUTA forms may still be required until progression. You can refer to Section 14.4 for further details. When reporting a second primary, verify with the treating physician that it is indeed a new cancer and not a metastasis of the existing disease. When documenting the new primary, it is important to use the date of histological diagnosis. Report if the patient received any non-protocol therapy prior to documentation of progression. Again, that is important for us to know, and we will be asking for dates of other treatment. If reporting the date of death, it is important to submit both a follow-up form and a notice of death form. And under the Notice of New Primary section, please specify the type of cancer for the site of new primary. For example, if the new primary is melanoma, enter that as the site. I don't think there's a whole lot to talk about other than that with the follow-up form. Again, it's a pretty standard form asking basic questions. And yes, we tell you that fields aren't optional. But again, if your answer is no to something, you don't need to provide further information. Again, if something looks a little suspect, explain away in the comments, and that's the easiest way to avoid a query. Moving on, the Notice of Death form, which you see on the right-hand side of your screen. Newer SWOG studies include the vital status form. There it is again. Please use the appropriate vital status, whether it be on treatment or off treatment. For this patient, it would obviously be off treatment. That version. Form to indicate a patient's vital status to have the notice of death roll out. So a lot of times in RAVE, you might be thinking, where's my off-treatment notice? My patient progressed, I need an off-treatment notice. RAVE is very much event-driven. You're gonna have to answer a certain question, yes or no, to get a form to populate, and it won't happen until you save that form. Just basic 101 RAVE. All protocols, if you go into CTSU in the master forms, you'll see, and forgive me for forgetting the title of the form, but it basically lines out all the forms for the study and what action rolls that form out. So if you're ever curious, like how do I get an off-treatment notice? You can call your data coordinator, in case I haven't mentioned that enough, 206-652-2267. But that form is really awesome too, because let's say you're, East Coast and we don't start till 6:30 here on the West Coast. So, if you want to that's your time frame, check out that form. It's going to tell you exactly what you need to do to get those forms. Yeah. I don't think there's a whole lot more to talk about with the Notice of Death form. It's very straightforward. It's important to note that if you just do unknown down everything, and submit the form, you're going to get a query. Again, this is a clinical trial. We're looking for data here, and we get it. Sometimes you don't know anything. And again, the beautiful use of the comments form field, put that in there. Hey, I tried this, this, and this. I can't get any information. Sorry, all I can provide is the date. We won't query you because you've proven yourself worthy. You tried. You failed. Not failed, that's terrible, sorry. You tried unsuccessfully. There are no failures here, I'm so sorry. So explain it away, and that will, again, avoid some queries for you. But keep in mind, it is crucial to obtain the most complete information as possible. They say that it's not sufficient to simply verify a death by accessing the Social Security Death Index or confirmation by the tumor registry. Okay. Every effort must be made to determine the cause of death and the corresponding details. Again, we are looking for information. This is a clinical trial. So any information, I think it was Elaine that said earlier, I don't think anyone's ever been penalized for giving too much information. This isn't like an FDA audit. This is a SWOG trial, man. We just want your information. If the cause of death was cancer, it's important to document which cancer. If it was the malignancy being followed in the study, tumor should be selected. If it was a different primary malignancy, new primary should be selected. A patient who dies of cancer most likely will have some evidence of progression prior to death, and it is important that the progression be documented. If there's no documentation found, the CRA should document the attempts to obtain it and the outcome of those attempts. Answering unknown, it's worth saying again. To all fields of this form is not a valid response. Unknown should only be used if all other avenues of investigation have been exhausted. If cause of death is truly unknown, use the comment section at the bottom of the form to explain all steps that were taken to try to find the cause of death. For example, was the location of the death certificate attempted to be found? Was the tumor registry checked? And has the family or primary physician been contacted? If unknown is selected, you will be queried. So that's just a more eloquent way of saying, explain it away to avoid queries. That's our Notice of Death form. Again, if you're unclear on how to complete the form, give us a call or an email. We're happy to help you out. I have learned over the years that you don't have a lot of time, and filling out SWOG forms, not your primary job. Sometimes we forget that in our beautiful world where all we do is SWOG, it's all SWOG all the time. We don't see patients, none of that. We look at your data and think, how did they not get this right? We're learning over time. There's a 100,000 reasons why you're not getting it right. We want to make sure that we're available to help you. And if that means taking 10 minutes of your day to say, hey, we just put patients on this GI trial, 1922. Can you just answer a couple quick questions so that when it comes time for me to fill out these forms, I'll know what I'm doing and I'll get it right, and then there'll be no more wasted time? We're happy to sit with you 10 minutes, 30 minutes. We've spent an hour on the phone with people just helping them navigate the SpecTrack system or the forms for a study. If you can make that time, we will always have that time. It is not our goal to write queries. It is not our goal to get snarky or go back and forth. We really want clean data, and we want to make that as easy as possible for you. This is SWOG, and as we've learned with other cooperative groups, and certainly the registration world, we're pretty simple, and we like being simple. So anytime we can be of help, guess what you're about to see? The list of DLs. We encourage these DLs because you might develop a rapport with me because I'm the only DC on the study you work on. But man, I take a lot of time off. A lot. I like it. I want to take more time off. Thanks, COVID. I've built up a lot of PTO. I'm ready to go. You don't know my schedule, and if I don't set my out of office, you don't know I'm gone, and your email sits and waits and sits and waits, and I pride myself on my response time. If you're ever waiting for me more than an hour, something's wrong. I'm on vacation or something, because that's not right. You use these DLs, it's going to go to the other data coordinators who do know my schedule. They'll be like, oh, Chris is out. She can't answer this, so I'm going to help. And sometimes the help might be, man, that's somebody else's study. Can you wait till tomorrow when she's back? And if you can't, they'll help you. But we know each other's schedules. We know to cover. So use these DLs. They will ensure that you get the quickest response. With that last final plea, this concludes this year's Practicum. Thank you for your participation today. We hope this has been an informative session to the best of our abilities, and that it will help you navigate the SWOG data submission process. Please take this opportunity to ask any questions at an open mic. Those mics are freezing cold, not used all day. Or if you're in the chat, for those attending virtually, we're gonna monitor it for at least 2 more minutes. Look, Kayleen is literally running to go monitor the chat to answer your questions. Again, this Practicum is an abbreviated version of the Practicum specifically made for our hybrid training environment. The full Practicum and all of the exercises can be found via the SWOG website under the training link. If you do have questions or concerns regarding any of the material in this Practicum, please feel free to reach out to the Disease Site Committee email distribution list for your study at any time. And of course, you may also call the SWOG SDMC at 206-652-2267 to speak to a data coordinator. Look, I don't know how I feel about this. We finally have our first question, and it's from an SDMC staff member.
Look out. This is a comment and a question, but first of all, the comment. It's an acknowledgement. A dear friend and colleague is celebrating her last Practicum today, and her name is Tracy Marr. I'd just like to give it up for Tracy Marr after 25 years of service.
She's right against the wall.
My question is directed to Tracy. They say after 25 years, I hear the first 25 years at SWOG are the hardest. Am I correct? Oh, okay. We don't wish you all the best. That's all, really. The last 10 were the hardest.
She said they were all wonderful online. I couldn't hear that. All of them.
That's it.
That's another thing really quickly. I know that in our line of work, we are losing a lot of people and having a hard time filling it, and so we're all being overworked and stuff. We're very proud that we're losing 2 SWOG members to retirement. That's the best way to lose them. It will be tough between her and Elaine, but we will muster through. But yes, thank you, Larry, and thank you, Tracy. Thank you to all of you for your time, your participation. I hope you enjoy the rest of the meeting. You're done for at least now, so thank you so much. Huh? Yeah, you can do it.
You were this close to getting out. The last thing I wanted to comment again was the evaluation forms in the back of your manual. Please complete those and drop them off at the registration desk right outside on your way out. Again, the certificates will be available to you online in a few weeks, and we'll let you know when those are ready. And I think when I mentioned it this morning, The audio is out for our remote attendees. There is a link on the SWOG website on the group meeting page for the clinical trials training course. It says evaluation form. Just click on that, it'll take you to SurveyMonkey, and you can submit your evaluations that way, and your certificates will be available with all the in-person attendees as well. So again, we thank you for your attendance. I hope you learned a lot, and I hope you reach out if you have questions either today or even after you get home. Thank you.
She needs a rest. The kids are playing up downstairs. Sister's saying in her sleep, brother's got a date to keep. He can't hang around.